This meeting is being recorded.
We have as the speaker today, an absolute expert without doubt, Dr. Kamini Walia, who is a scientist at ICMR. And she will deliver the talk on the topic of encouraging fundamental research and translational research in AMR, and most of her talk will focus on diagnostics. Most of her work has been in the diagnostics space. Dr. Kamini Walia is a microbiologist by training and has also completed her master's in public health from Johns Hopkins. Currently, as I mentioned, she is a senior scientist, Scientist F, in the Division of Epidemiology and Communicable Diseases at ICMR, and leads the Antimicrobial Resistance initiative of ICMR, which focuses on various aspects, including surveillance, antimicrobial stewardship, and the One Health aspects. Prior to ICMR, she was Director, Research and Development at PATH, and a former member on the WHO WHO Scientific Advisory Group of Experts on Essential Diagnostics, and Commissioner on the Lancet Commission on Diagnostics. Her experience is diverse and quite wide, spans working on infectious diseases, including HIV and AIDS programs, and health technologies, including vaccines and diagnostics. She is a recipient of ICMR's Shakuntala Amirchand Award and the Indian National Science Academy Young Scientist Award. She has received numerous fellowships and training from WHO, NIH, IVI, and the Pasteur Institute. This talk is being organized by CCAMP as part of the CCAMP, CARB-X GAN accelerator network. CCAMP, as most of you know, is part of the CARB-X accelerator network, the only one in outside of the US and Europe. Just wanted to say that CARB-X provides scientific and business expertise and support. There are 6 global accelerators around the globe. As you can see, CCAMP is the one that is the only one outside of US and Europe. And these accelerators support the development of new antibiotics and other life-saving products to fight drug-resistant bacteria. So under this, our association with CARB-X, we are bringing this webinar to you. Just a disclaimer that the content of this presentation is supported by CARB-X. CARB-X's funding is sponsored by the cooperative agreement number, ID SEP 160030 from ASPR BARDA, and by awards from the Wellcome Trust, Germany's Federal Ministry of Education and Research, the UK Global Antimicrobial Resistance Innovation Fund, the Bill and Melinda Gates Foundation. The content is solely the responsibility of the authors and does not necessarily represent the official views of CARB-X or its funders. Just wanted to have this in place. With that, I would request, before I just hand it over to Dr. Kamini, just like to welcome all of you again. I have my colleagues, of course, Dr. Taslim Arif Sayyed is the CEO and Director of CCAMP. We have Dr. Swati Subodh, who's the Science and Policy Lead at CCAMP. Chandrika and Meghna have also joined the call, and I also see Titash, who's from our comms team. Just want to, and Dr. Walia, over to you.
Good morning, everyone, and thank you, Nandita and Taslim, for this opportunity to share my thoughts on from my experiences of working in the AMR space till now, and how I see the landscape to promote the fundamental as well as the translational research. Having worked in the AMR space for last 7-8 years, I will be sharing my thoughts around some of the examples that I have seen, which can actually enlighten the audience on what kind of challenges are there in translating some of the new drug leads or the new diagnostics, and how to navigate that space. Although I may not be able to provide all the answers by end of the session, but I will try my level best to share with you the experience so far. So I will just share my screen now. Please confirm that you are able to see my slides in the slide mode.
Yes, Dr. Kamini.
Yes, Dr. Walia, you can.
So thanks once again for this opportunity, Nandita. And just giving a broad overview of why should we be worrying about antimicrobial resistance at all, because antimicrobials, which includes a broad range of drugs, antibiotics, antifungals, antivirals, are actually the foundation of modern medicine. And the trends and the patterns of the drug resistance that we are seeing in our country are actually very alarming, and they are a direct consequence of excessive use of antimicrobials. Globally, they are threatening the advances which were made in the control of infectious diseases, and will also impact 18 targets across the 9 sustainable development goals towards achieving good health and well-being, eradication of poverty, providing quality education, clean water and sanitation, and many others if we don't address it today. So India is one of the countries which has 18% of the world population, and we also have highest burden of drug-resistant infections in our country. And most of the burden of AMR is actually in the low middle income countries. And unfortunately, the data that we have, or the interventions that we have been developed by high income countries, because they have the data and they have the means to implement it. So it's a long way to go for India in terms of containment of AMR, yet every single step counts. Bacterial infections which are untreated claim almost 700,000 lives per year, and this figure has now been updated by the recent Gram Lancet report, which shows that AMR's morbidity and mortality is even higher than what is claimed by HIV, TB, or malaria together. So Jim O'Neill's report in 2014 said that it will cross almost 10 million people deaths per year by 2050. From the study which we carried out in ICMR, we have found that the cost of treating drug-resistant infection in government hospital was 38% higher than the cost of treating non-resistant infections. This is unpublished data yet. What we have seen in the past is that in India, many simple clean procedures like a cesarean section or any cardiac related surgery are being complicated by hospital acquired infections, which are mostly drug resistant. And in the COVID patients, we found almost 50% mortality in patients who had drug resistant infections, especially Klebsiella pneumoniae. So the way I like to put it, these are invisible threats which claim invisible victims. The reason I call them invisible victims is because no system is actually recording how is the morbidity and mortality impacting the lives of the patients. So they continue to remain invisible to our healthcare system. There are numerous factors which actually create selective pressure for development of drug resistance, which includes many factors related to our compromised healthcare system, like poor doctor to patient ratio, limited diagnostic and laboratory support, lack of ID physicians, physicians preferring to use broad spectrum over narrow spectrum antimicrobials. And then there is a patient behavior like self-medication, non-compliance, and also somewhere I think increasing affordability of antimicrobials is very important. Then we have high 12.4 billion pharmaceutical industry, which also manufactures a lot of antimicrobials, and these antimicrobials are available without prescription, most of them. Only for some prescription clause has been introduced through the H1 schedule, but then there are also concerns around counterfeit drugs and inappropriate FDCs. So the whole situation is very conducive to development of resistance, and besides that, there is a excessive use of antimicrobials in livestock and poultry, which I will talk about later. The consequences that the patient is that the receiving end of it, I mean, the mortality is high, then there is high cost of treating drug-resistant infections, which not only lead to individual patient losses, but also losses to the state in terms of loss of productivity and others. Now, this is the One Health component of AMR. The human antibiotic use has jumped by 36% in the year 2000s. Up to 75% of the antibiotics are used in aquaculture, and 70% of the antibiotics are used in the animal sector, and it has jumped by 67% in last few years. So all this is actually excreted through the urine and feces. And it also finds way into the drinking water. It finds its way into our rest of the ecosystem, and through which it enters food chain. Now, this is something that we have been able to track. But how much is the public health impact or the clinical impact of this kind of dissemination? That still needs to be understood. And that's where it's very important to support research in this area, that how much is the antimicrobial residues in our environment, and what is their clinical and the public health impact. So AMR containment, if we see broadly, includes improving evidence, improving the diagnostic stewardship, so that correct diagnosis actually guides the use of antimicrobials, and not this syndromic approach which we have been following in our country because of lack of laboratory support or absence of diagnostic support at all levels of healthcare. And the most important component is the infection control here, because we are a country with a very high microbial burden. Whether we talk about community or we talk of hospitals, the sanitation and hygiene is poor. So to compensate for poor hygiene and sanitation, the antimicrobials are provided to the patients. In a hospital, if a patient is admitted, sometimes they get more than 3 or 4 antibiotics, which also includes antifungals, because the doctor doesn't trust the infection control processes of the hospital. So we cannot exercise antimicrobial stewardship unless the infection control processes, both in the community and hospital, are improved. Only then responsible and rational antimicrobial prescriptions can be practiced. Till then, the suboptimal stewardship around antimicrobial prescribing will continue to increase the antimicrobial pressure. All this is reflected in the National Action Plan on AMR. So these are the 6 pillars. I would be focusing more on the 5th pillar of AMR for rest of my talk, which is that how important is the innovations, the research and development, if we really want to address the problem of AMR in our country. This has, as you can see, 3 components: the new medicines, diagnostics, innovations, and financing, which is the most important pivot, and nothing can really move forward without financing. Now, as many of you may know, that ICMR has a network of hospitals through which we capture data in tertiary care hospitals, and we publish these trends annually. What we see is that our hospitals have a very high burden of gram-negative infections. E. coli, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas. They continue to be the most isolated pathogen in these hospitals, with very high ESBL rates and carbapenem resistance. The carbapenem resistance has seen consistent increase in last few years, because there's another drug in the market by name of ferropenem, which has cross resistance with carbapenems, and which is given orally. So whenever a drug is introduced as an oral product, the misuse of the drug is very high. We are also seeing increasing colistin resistance. 40% of gram positives are MRSA, who are also showing MICs, increasing MICs to doxycycline and linezolid. In fact, linezolid resistance has increased a lot in last few years. 1.6% of the total isolates are vancomycin resistant. Both vancomycin sensitive as well as in vancomycin resistant, they are resistant to linezolid. Vancomycin resistance is 3 times higher in Enterococcus faecium and faecalis, and this has increased in last few years. Glycopeptide resistance also has jumped from 15% to 30% over last 2 years. Phosphomycin resistance was encountered in 11% of the enterococci isolates. We are also seeing 30% resistance to caspofungin and fluconazole in Clostridium glabrata and Candida glabrata and Candida auris, because more and more labs we are seeing now are concerned about the fungal infections as well. So this gives you a broad idea of what kind of drug resistance profile we are seeing in pathogens which are of clinical importance to us. I forgot to mention the S. typhi. This, I think, is the only bright spot which gives us some hope, because in 1990s and early 2000, we saw a lot of MDR Salmonella typhi, which was resistant to ampicillin, chloramphenicol, and cotrimoxazole. But then as the use of these drugs declined, we saw 100% sensitivity to these drugs, but at the same time, we are seeing increasing resistance to ciprofloxacin, which is now rampantly used for treatment of typhoid. And why it is used so blindly, that also I will come in the later half of my talk. We also track the drug the genotypic characterization of antimicrobial resistance in these pathogens, and we see that the drug resistance, the genotypic profile is very different in our country. We have more of NDMs and OXAs compared to KPCs and other genes which are predominantly found in other countries. So that's why the fundamental research, which includes the basic research, has to go on so that we are aware that what kind of changes are happening in the pathogens as they circulate in our community and in our hospitals. This will also, I think, give us an opportunity to forecast how things are going to change if we expand these efforts to other areas like the environment and the animal sector, so that it is able to give us we are able to give a forewarning to our clinicians as to what they should be expecting 6 months from now or 1 year from now. As a country, we use a lot of broad spectrum antimicrobials, which includes piperacillin, cefporazone, sulbactam, carbapenems. Almost 50 to 75% of the drug use happens from the watch and the reserve category of WHO, and we prescribe this is data from the COVID patients, and they received only 16% of the drugs from the excess list of WHO, which is a narrow spectrum antimicrobials. Rest all this is broad spectrum antimicrobial, and also a lot of BLI, something like an augmentin, something like piperacillin, tazobactam, extensive use of these drugs. And this is actually a symptom of poor reliance on laboratory support or diagnostic stewardship. That's why it is very important to have good diagnostics. This is just a trend, what we are seeing in COVID and non-COVID patients. Klebsiella pneumoniae has become very difficult to treat, and as you can see, it has jumped. The MDR Klebsiella pneumoniae has jumped from 60% to 77% in our network, and similarly, E. coli, Acinetobacter baumannii is having horrible, very high drug resistance levels, and MDR Acinetobacter is now almost at 93%. Pseudomonas aeruginosa remains less than 40%. But this is where it is very important to track the genotypic profile of the bugs. And what we are seeing is that even the drugs which are in the pipeline or are likely to be introduced very soon in the Indian markets, they will not be effective against many of the genes which are currently circulating in Indian hospitals, which include the NDM, OXA 48, 23, 24, and 58. Then so many of these drugs. Uh, and we also have a lot of carbapenemases against which many of these drugs will not be active. So that's why it has become very important that research in antimicrobial resistance be made a priority. It is important to understand the drug resistance mechanisms, clonality, and transmission dynamics, how it happens between the human, the livestock, poultry, and the environment. We also need new drugs for our patients because of the increasing levels of resistance to the previously being used drugs. Identify new drug targets, identify new delivering mechanisms, and of course, most important, and what I am very concerned about, is having good diagnostics so that whatever we have currently can be used judiciously. And that is only possible if there are good diagnostics to guide antimicrobial prescriptions. Then there is a whole new area of repurposing antibiotics, the ones which we had forgotten before, and how new combinations can help improve the treatment. In fact, anti-infectives and infection control is a relatively unaddressed and very ignored sector. I mean, there are interventions available in this area, but then we really don't have mechanisms. At least I have not seen in my career, my experience in AMR of testing any new interventions which help reduce infections in hospitals, which is also again very important. And then of course new mechanisms to how we can monitor prescription behavior, the patient behavior, and other infection control processes which are there in the community. So as the topic of this talk today is how we can encourage fundamental and the translational research. First, it is important to understand why do we need this research in first place, because a lot has been done in the past. There are many products which are currently available or being researched in the European or the Western countries. But why should Indian effort be so important? Because the first and the foremost reason is that we have the highest level of antibiotic resistance. As I mentioned in my previous slides, antibiotics like meropenem are considered last resort, but they are used extensively. As a result, most of the pathogens which are treated by these drugs have become resistant to these drugs. The AMR, unfortunately, has not drawn required attention for meaningful and actionable action, as well as resources in terms of its containment, and especially discovery of novel antibiotics. This is still done in an academic mode by research institutions or universities, and with the completion of the project, the PI rarely knows where to go, and if he has or she has discovered a useful product, how this should be taken forward. And I think that is the biggest, I would say, bottleneck, because of which we are not able to harness the full benefit of the research which is being done in research institutions of our country to our advantage. It is also a fact that all major pharma companies have exited this research area several years ago, and essentially countries like India, who have a high rates of drug resistance, have been left on their own. Our healthcare systems are different. We have a very strong private sector, and we also have a public sector. And because of this complexity, and sometimes or rather most of the time, since the public healthcare infrastructure has not been augmented very well because of the low investments in healthcare sector, we have several limitations under which the lab has to operate, or a patient has to be treated. That makes the needs of our healthcare system very specific. And we have also realized that some of the devices or diagnostics which have developed, and even drugs which have been developed in high income countries, or the countries which have a very different drug resistance profile as well as a very different healthcare system, may not really address the needs of low middle income countries like India and many others. So promoting research in AMR basically requires your infrastructure and resources. It can come from public and private funding. Robust surveillance has to continue and provide updated database and evidence of what is actually happening and how are the pathogens evolving. Then there has to be a suitable translational process so that there is a faster approval process for AMR diagnostic R&D. There should be more discussion around IP protection and cost incentives to do the same. Then there should also be supportive policy which actually incentivizes doing research and development in AMR. And then there are operational factors which like stakeholder participation and multidisciplinary R&D efforts. I will be discussing most of them in detail in next few slides. So what do we really mean by fundamental research for antimicrobial resistance? This is a combination of the clinical and the basic research. We want to understand how bugs are evolving, what kind of new drugs and treatment modalities would be needed to address the infections caused by these bugs, what kind of diagnostics and infection control interventions can help support the overall agenda of responsible antimicrobial prescription. And what we really need is good quality diagnostics, devices and drugs as a good outcome from this. But what we essentially require to support it is a very vibrant scientific community which is willing to take up these challenges, and this community has to be supported by adequate funding. The most important area I think is which has been neglected so far, although the National Action Plan on AMR addresses it briefly, is the consensus on what are the priorities for the country. And there was a priority listing which was done in AMR. There was a priority listing which has been done by ICMR. I'm sure there's a priority list with the DBT and CCAMP as well. But then how do these priorities align? And so that's why it has to be, the collaborations needs to be strengthened across all the agencies and the key partners and stakeholders who are working in this area, just to avoid duplication, because with the limited resources that we have, we really can't afford any wastage. Other than that, I think the technology is changing very fast with the machine learning and the artificial intelligence coming in. And I'm yet to see good initiative coming from India, where how ML and AI introduction can actually help us understanding what is going on, strengthening the evidence as well as the forecasting, because that's what it boils down to. Are we able to preempt the threat and prepare ourselves for that? So that's something that needs to be done. So the way I see it is that, it is going to require a more cohesive and a collaborative effort between the biologists, the engineers, and the clinicians as we move forward, because unless we do that, it is not possible to come up with a good product which addresses our requirements. Now, the translational research is effective utilization of research products and innovations, and it requires undertaking field feasibility studies, clinical trials. In order to support that, there should be structures and processes for translation. Most important is, which we forget sometimes, is the translational cannot happen if there is no demand for the product. Unless, and this we had seen in many diseases and their containment in the past. For example, malaria diagnostics could not have processed, could not have been advanced at this stage unless there were not demands by countries and the programs for a good diagnostic product. And that's why how we are using now RDTs, which are very good for malaria detection. Again, the funding and the policies needs to be supportive of this. But what is most important is the structures, the regulatory structures which move seamlessly and a supportive industry. Out of all these, I would say the regulatory structures are most important, because unless you have a structure and a process to move things from one stage to another, which is streamlined, which is seamless, and where the roles are well defined, there will always be challenges and there will always be roadblocks, which will eventually lead to sometimes wastage of a very good devices and new diagnostics or the new drugs. Now, why is new antibiotics an urgent national priority for us? Few antibiotics which have been developed or discovered in the past, they have found way to India in a timely manner. As a result, our patients have to wait for almost 5 to 6 years for novel antibiotics like daptomycin or ceftaroline to be introduced after their initial launch in US. Similarly, antibiotics like imipenem relebactam and meropenem vaborbactam, which are now available in US for more than, and European Union countries for more than 2 years, are yet to be introduced in India. So on an average, the period between the novel antibiotic being used in Western countries and between their introduction in India, the time period is about 5 to 6 years. As a result, our patients are left to suffer the serious infections. Sometimes they are given some cocktail of multiple antibiotics, which we don't know how good is their performance and what are their side effects. Yet, because of the limited options which are available, clinicians try with everything, what they can to help their patient get better. And we also saw a recent shortage of ceftazidime avibactam, which overcomes only a part of resistance mechanism prevalent in India and actually left many sick patients in lurch. So the doctors in our country, since I work very closely with them, have been requesting unhindered availability of imported antibiotics. The new drug development, there's nothing which has been introduced recently. The only new drugs introduced are mainly the analogs. But then there are no new class of products which has been introduced in last few years. Although there is now evidence which is available in the surveillance system. But one more thing, which the question which I get asked many a times when we participate in international meeting is, what is India doing about the stewardship? Because unless we have good antimicrobial stewardship in place and good regulatory stewardship in place, these antibiotics also, the newly introduced ones, will meet the same fate, that they will lose their efficacy in 3, 4, or 5 years because of their rampant use. ICMR has funded few projects in the past which have looked at restoring the activity of available antimicrobials. So these have been funded in different institutions. There's one in IIT Roorkee, there is one in CSIR Institute, CDRI. So these are different metallo-beta-lactamase inhibitors, and they have reached the patent stage. For example, one, the provisional patent was underway till few months back. I'm sure they have received it now. But then subsequently, these research efforts really wither away and don't reach anywhere. We do have a success story in India of cefepime zidibactam drug. This was discovered in India, and it is a combination drug effective against wide range of MDR and XDR bugs. The studies independently conducted by leading India, US, and EU investigators established that this possesses India-specific features of broadcast coverage of highly resistant pathogens like Klebsiella, Pseudomonas, and Acinetobacter, and even US FDA granted Quid P, which is the qualified infectious disease product status to this combination product, and this was entirely Indian endeavor. So having seen these kind of successes, ICMR is also working with the CARB-X, which is the Global Antibiotic Research and Development Partnership, and we are working towards developing new drugs for which can be helpful in treating serious bacterial infections in India, whether they cause sepsis or whether they cause any other syndrome. CARB-X actually has it's a not-for-profit organization, and it is developing few new drugs which will be tested in India. And the advantages of doing the clinical trials in India would be that these drugs will be available to Indian patients earlier than what our experience has been with the new drug products in the past. Now, talking briefly about how AMR diagnostics landscape is evolving in India, and what were the challenges that we found in a small project that we did in the past, which we also published. As I mentioned previously, diagnostic stewardship is the most important pillar of antimicrobial stewardship. For now, mostly, especially in the public sector system, there's a lot of reliance on the blood culture or the urine culture. Many of the labs even don't have necessary infrastructure to deliver that. So that makes the point of care and RDTs, the rapid diagnostic tests, which are developed in India, which are affordable and which will address the needs of the Indian patients in healthcare system, very, very of paramount importance to us. There are many diagnostics which are available by many multinational companies, but they are sometimes unaffordable and sometimes don't deliver what they are expected to deliver in our kind of climate and our kind of laboratory situations. Now, case in example here I would like to mention is the typhoid. Typhoid is a disease which is of very high importance in India. We have seen the resistance developing to chloramphenicol. There have been MDR outbreaks. Now we are also seeing ciprofloxacin failures and also creeping MICs to cephalosporins. These are the three drugs which are currently being used for treatment of typhoid. Yet we don't have any good rapid or a point of care diagnostics. The ones which are available, they have limited sensitivity and specificity. Blood culture remains the gold standard, and the blood culture is actually most of the time negative because the patient has already taken an antibiotic when it comes for the when he or she comes for the blood culture. So now this is something, the reason I pointed it out here is that this is a challenge which is exclusive to India. Typhoid is and also Southeast Asia region. But then in spite of having the disease for decades almost and seeing how it went from MDR to non-MDR to a new profile of MDRs, which includes ciprofloxacin, cephalosporins, and azithromycin, this kind of outbreak we saw in Pakistan a few years back. But then still there is no point of care diagnostic which actually fills the gap of providing good diagnostic and subsequently antimicrobial stewardship. So while I'm coordinating the activities of antimicrobial resistance in India, we were concerned about what all activities are being undertaken towards development of point of care diagnostic or rapid diagnostics, which can be useful for containment of AMR. Having participated in many of the meetings organized by BIRAC, by DBT and DST, I had a fair understanding that a lot of funding has gone into supporting many efforts which for development of diagnostics which can have potential for antimicrobial stewardship in India and other low middle income countries. But to our surprise, we basically what did was that we sent out a questionnaire to some of the known innovators and developers who we knew had a product as to what were the challenges that they were facing in introducing their diagnostic into healthcare system, something like a national health mission or any state healthcare system, or even introducing them to the private market or the government. What we found was that 1 minute. So what we found was that there were no target product profiles for priority syndromes in the country. So the innovators and developers really lacked guidance on how they should be developing a new diagnostic. And once the diagnostic was developed, although we do give credit to the developers that they all had some clinician or microbiologist included in their team, and most of the developers were coming from the engineering or the non-medical background. Still, they had the foresight to include a clinician or a microbiologist, which I think led to development of good products. So the products that they had developed, they could be useful if they are able to overcome all the challenges and hurdles and make it to the National Health Mission. Then there was no validation protocol which are readily available for these developers, which they can follow in terms of systematic evaluation of the tests. So a synchronized process basically, which helps them overcome the bottlenecks and facilitate expedited market introduction and clinical uptake was essentially missing. Now, I will just some of the factors which actually led, which we found were the main key bottlenecks were these: the absence of target product profiles. There was absence of gold standards for comparison. For example, if there is a test which has been which will detect sepsis in saliva, how do you compare it with what gold standard do you compare it with? You can't be comparing it with the blood culture. This was brought to our notice by one of the innovators. Then we also realized that there is a long duration of development process. There are absence of clearly defined process for validation of indigenously developed antibiotic. Till many years back, what was happening was that most of the diagnostics were being imported. So there was a guideline that if certain test is to be imported, you import a limited number of tests, do some sensitivity and specificity in maybe 100 odd samples, and you were given the market authorization to market the product. But what I realized was that the system is not really prepared for handling the indigenously developed diagnostics. And that's why there is no guidance which is available, even to undertake the field feasibility studies or the validation studies. Although none of the developer mentioned to us that they had any shortage of funding, but the development process takes about 8 to 10 years, and there is lack of funding to support all steps of validation, because there are certain delays because of misinformation or the innovator not understanding the next step by the time your previous grant finishes, and then for the next step you have to find the fresh grant. So these were some of the challenges which we found the innovators and the developers were facing. This is one of the tests which we have identified, which ICMR will be doing the field feasibility with. This is a test which allows identification of gram-negative and gram-positive pathogens. It allows detection of phenotypic identification. And also, it has been developed by Dr. Suman Kapoor in BITS. And this is an excellent example of what I was referring to in my slides, that BITS has a curriculum which allows for training in engineering with biological sciences. And it is from this speciality that the tests like these have been developed. And of course, there are many such examples in the private sector or in the startups where the engineers are working with the clinicians. But this, if we really want to go very far, this is something which we need to encourage in our academic institutions and research institutions, which is a collaboration, a collaboration of multidisciplinary teams, and each of them brings their own skill. So this is briefly the project that is currently ongoing. We are looking at development of validation protocols. We are looking at development of target product profiles for Indian diagnostics. We have developed one for sepsis. We have developed one for typhoid as well. We will be also undertaking cost effectiveness studies of these diagnostics, because the major criticism of using diagnostic is that diagnostic is more expensive than the antimicrobial. Now that is something that we have to get over for our systems to invest more in diagnostics, if we are able to document that what will be the long-term gain of investing in diagnostics. So we are in the process of doing that. Just briefly, I would like to focus on the target product profiles, since many of the participants in this audience will be innovator and developer. Please look for target product profiles whenever they are available for a drug or a diagnostic or certain product, which may be available from, sometimes available from WHO, sometimes they will be made by your available by your own country. Because if you don't have a target product profile, you are essentially a misguided missile. You are going in every direction but not really knowing what you want to achieve. So to have an optimal design, to have an optimal a design that leads to maximum efficiency, you really need target product profiles. And there are many such which are available through different agencies, which can provide you the basic information. So TPP is essentially a strategic planning tool. It can assess potential pitfalls, as well as help you create mitigation plans at all stages of clinical development. They can aid in planning through distribution to clinical and non-clinical research organizations in order to solicit advice and modify the existing plans. And also they are helpful in promoting the team-based approach. It can minimize the risk of late-stage drug development or a diagnostic development failure. They increase the probability and optimal safety and efficacy data, which are so that they are available in the timely manner. They also allow for improved label content, as well as decrease the total amount of time spent on entire drug development process when you have this kind of a guidance earlier on, which is available to you. Now, this is I think the most important slide of my talk here. This is what, from the diagnostic perspective, this is what the landscape looks like. There are many funding agencies which are funding different research efforts, which include DST, ICMR, DHR, DBT, BIRAC, DSIR, DRDO, and the CSIR. And then there is a lot of private funding which is available through individual donors, companies, and NGOs, not-for-profit organizations, which the entire innovation in the country is funded by this. But then much of this is happening in the research institutions. If they are able to end up at this stage, diagnostics developer stage, with the startup or with the company, the next step for them is creating a data for validation of a diagnostic, which includes the analytical, clinical data, shelf life, toxicity, and all other necessary data fields need to be completed. Now, once that happens, most of the diagnostics are stuck at this stage. And even they don't know whether the data is satisfactory enough to be going forward to the DCGI approval, or they would need additional data for looking at the field fitness, cost effectiveness, the issue of scalability, etc. And this is where ICMR is trying to help some of the AMR diagnostics which have been developed till now. Now, there are many other instruments which have been created. There is a National Health Innovation Portal. This is portal under the National Health Mission. So National Health Mission is also looking for new diagnostics and devices which they can introduce through their program. But the requirement is that once the innovator has completed this, it can upload its data into National Health Innovation Portal. And then if this portal finds it suitable, they will subsequently introduce it into the program. The other such instrument which exists is the HTAIN, which is the Health Technology Assessment Board under DHR. This also has expert group which recommends diagnostics and takes them to the uptake. Then there is a regulatory agency. If everything is found satisfactory, they can be approached and they can subsequently suggest it for the uptake in the healthcare system, or even specifically for any disease specific program. Now, the crucial factors here is the independent evaluation of the test performance, field feasibility, cost effectiveness, and the evidence of scalability. Subject to this, the diagnostics move forward. But then there are a lot of challenges here. This health innovation portal, many of the innovators who we contacted, they had never heard about it. Health technology assessment board's role is also a little unclear as to how these two agencies actually replace or augment or supplement each other. So a lot of clarity actually is needed even within this landscape. But the only, I think the positive side here is that at least there is now recognition on the part of the government to be receptive to the indigenous and the innovative products which are being developed in the country, and there has been an attempt to create the pathway. Now we will continue to work with all these agencies so that this pathway works smoothly. But as a push for, I think, Atmanirbhar Bharat or Make in India, there has been a good recognition of innovation within the country, and I'm sure within the next few years, these pathways will also be more smooth and more easy to work around. Now, this is the validation protocol that we are developing. This is still under the development stage. What we are aiming to do is that we provide a validation process to any innovator or a diagnostic developer, which he or she can use to achieve desirable outcome and decision, so that this improves the assay usability and also does away with the barriers to its implementation. Now, for this, we are in discussion with DCGI, because whatever ICMR proposes, it can be utilized only if the DCGI also endorses this validation protocol. We are still in the process of doing that, but once this protocol comes through, I'm sure this will help many diagnostic developers as to how they should be navigating through this very complex landscape. Briefly, I will touch what is the regulatory environment for new antimicrobial drugs. As many of you would know, DCGI is the nodal agency for undertaking this research. For giving approvals for new drugs. There has been a lot of transformation within the regulatory landscape in all these years, and now there is a reduced time for applications. There are 30 days for the drugs manufactured in India and 90 days for those developed outside. A lot of responsibility has come on the sponsor as well as on the ethics committee. There are now stringent guidelines for ethics committee to monitor all trials. New drugs approved for use in selected developed markets can be automatically introduced in India if there is a need to do that. 2 drugs for TB program have been introduced in this manner. There are new rules to remove regulations on tests conducted in animals in case the drugs approved and marketed for more than 2 years in well-regulated overseas market. So this is all for introducing new drug. For new drug approval, basically in India, these are the steps that you have to follow. The application has to be submitted along with the data of chemistry, manufacturing, and animal studies. Copy of application must be submitted to ethics committee, and clinical trials are conducted only after the DCGI and the ethics committee approval. They should also determine the maximum tolerated dose in humans, adverse reactions, etc. So there are different phases that you could do, the phase 1, phase 2, and phase 3, depending upon how much research has happened in the other countries, or if you are starting de novo in India, you could do phase 1 in India as well now. There are new drug registration rules which are applicable to completion of trials. The comprehensive information on marketing status of the drug is also required on information and safety, which is also comprises the phase 4. So this is what the process in the CDSCO looks like. There are provisions for accelerated approvals. The clinical data requirement may be relaxed under certain conditions. The subject expert committee now has experts from various areas, and permission and rejection to be informed in 90 days, which is very, very time efficient, I think. So this I have already discussed. So what can we really do to strengthen the fundamental and translational research around AMR in India? There is a existing infrastructure which has been created through different mechanisms, different agencies, and there are capacities and technical know-how which can be built upon. But what is very important is creating global network of experts, advisors, mentors, and trainers who can guide us because they have implemented this process at their end. Engaging the end user sometimes, which we forget, is very important. Investing in target product profiles, then also updating the ethics, intellectual property, and the regulations, and finding the novel funding mechanisms, how we can fund these activities. As I mentioned previously, identifying national priorities will be helpful to all the agencies. Having cross-cutting discussions, whether it is academia, industry partnerships, or multidisciplinary partnerships, basically innovating through engagement and also at the same time finding mechanisms to translate, but nothing to beat the blue sky research, which is the larger landscape of research from where most of our interventions will come from. And basically using the research to bridge the gap between the developer and the user, and reducing the time for that, creating guidelines for national standards and common standards and vocabulary for technology which are understandable across multiple disciplines, with an overarching aim to make technology available and affordable, not just for individual benefit, but also for the national programs. I'm sure you all would have seen this very big grant coming to IISc Bangalore, and with an overarching aim to strengthen the innovation and the clinical research. I think this was setting Twitter on fire last few days. And what I also found very interesting was their plan to introduce the MD PhD and MD MTech programs, because now there is good amount of recognition that we cannot venture into clinical research and advanced diagnostic and research facilities without having the multidisciplinary approach to it. So this is my last slide, and once again, thanks for this opportunity, and I'll be happy to answer if there are any questions. Thank you.
Thank you. Thanks a lot, Dr. Kamini Walia. I think you really covered it, I would say, end to end. Starting with laying out the whole landscape in terms of, why surveillance and bringing in the stewardship angle. So really relying on how, what one needs to sort of refer to while, wanting to develop a product. It cannot be, without all those initiatives. And then actually, coming down to, specifically if you want to develop a diagnostic or therapeutics, what are the steps and who are the agencies and the thing. And I think it's also very good to know and hear that there is a constant listening from the government agencies to, be very proactive to, enable this. Otherwise, we lose sight of that very often. So I think thanks.
Many things became clear to me while we were researching for our the AMR diagnostics.
The BITS project. Yeah. So you probably in that way wore a entrepreneur's hat yourself in that sense at that time. So if there are questions, I think what I would suggest is maybe they can unmute and identify themselves and ask the question. But there is one question which is on the chat. So I'll just read it out. But in the meanwhile, probably others can think. Dr. Kamini Walia, Dr. B. Pachayappan wants to ask your thoughts on how much of scrutiny a molecule shall undergo before being approved for any kind of research or manufacturing, or how we can scrutinize already approved antibiotics to prevent AMR.
Let me read it. It's a complex question.
Yeah.
So for antimicrobials, there is a clear guideline which is available. I understand that, how much research we have to undertake. I think the first step should be the proof of concept that it works and it doesn't cause excessive toxicity. So once you have these two things, then you can go to the animal toxicity studies. Then you can go to the other steps which I had mentioned in my in my presentation. What all you really need, which is 1 minute. Let me. So the first. So you need data of chemistry, the animal studies, as well as the toxicity studies, basically, to and the proof of concept that it works. So that kind of data is then sufficient to take you to the next step. So the animal toxicity is followed by the bioavailability and the bioequivalence studies, and then you are ready for the clinical trials. I'm sure someone like Anand, who CCAMP work very closely, he can answer all these. I mean, how many steps they had to follow before they actually went to the phase 1 clinical trial stage, which they are doing currently.
Yeah. So Akshit has a question. Akshit, you can unmute yourself. Maybe you can just say what you're working on and ask your question.
Good morning, everyone. I'm Akshit, co-founder at InVisio Biome and also working as project associate in Northeast in Dr. Shmin Chauhan's lab. So we are studying biofilm formation as AMR mechanism using in vivo animal model. Since more than 80% of human infections that we know are associated with biofilm formation, lifestyle of microbes. So the preclinical platform that we have provides uh, wonderful insights to know effectiveness of antimicrobials, coatings, and even vaccine development. So, so I would first appreciate and thank Dr. Walia for presenting a serious and a very informative case. We are so happy that our research and so well fine-tuned to address AMR. Currently, our team is working with pan-drug resistant clinical isolates of Klebsiella pneumonia, coatings on medical devices, and even using sponges isolated from hospital sewage water to treat pan-drug resistant isolates of Klebsiella pneumonia. So having leads on therapeutics, preventive strategies like coatings on devices, and even leads about vaccine development for escape pathogens, uh, we are fortunate and at the same time at a juncture that how we can shape these critical leads because they are already under peer review for communication. So we don't want to stay there. We want to reach out to the hospitals. So, and we are also thankful to the ECD, Dr. Walia, for even considering support to our lab to ad hoc ICMR program last year. So, like it took 4 years for our lab to get money from government agencies. So right now we are looking for more private investments, venture capitalists, so that we can hire more technicians, scientists to help validate our leads, which are already at preclinical levels and take them ahead. So maybe if CCAMP can help us a whiteboard session with Dr. Walia, we can learn more from her guidance and advice. So that would be great. Thank you so much once again, CCAMP and Dr. Walia, for the wonderful session. Yes.
Thank you. Yeah, but it again brings us to a point as to where do drug developers, molecule developers, basically like him, Akshit, go once they have a proof of concept. So for that, I think CCAMP, since I'm more focused on the diagnostics field right now, if that can be, if we can identify the stepwise process for new drug developers as to what they should be following, I think that will be very helpful to many. Because as I said, many such research leads originate in research organizations and just disappear from there. For many years, even during Dr. Bhan's time, there was a lot of discussion on research to translation. But then somehow things have been slipping through, and if CCAMP and ICMR can definitely work together to identify these next steps. one of the challenges I see in our country is the absence of the support to do animal studies. Most of these developers have to go abroad to do animal studies. I don't know what is the situation now, right now, but this was the
Still
situation.
that way, I think. It's still easier to do it
because
outside.
all the research related to animal in biology was stopped because of one politician activist we all know. So somehow I think, and that I think is the biggest challenge to many innovators and developers which I have, with whom I have had any interaction, because animal studies abroad are very, very expensive. And that's the reason most of the developers, even if they have a good potential product, they are not able to move to the next step.
Sure, I think we can certainly look at something maybe on the lines of the HTA, or I don't know, some mechanism we can work on that.
So there are a few
thanks.
more questions, I think.
There are a few more questions, yeah. There is
from
actually
Ankita Arora. She's asking whether it should be devices, diagnostics, or drugs. Well, if I have to identify one priority area, I would identify diagnostics, because the new drugs are not going to come very soon. Drug development is a very intensive process. It requires extensive funding. It requires a lot of time, and it is not easy to come up with new drug targets and new drugs. So in that case, it makes more sense to utilize judiciously what we have. So improving the diagnostics availability and improving the infection control processes is very, very important. And between the two, I will again pick diagnostics. That is something which is a low-hanging fruit. Plenty is available. It is on government's radar to improve the diagnostic availability through free diagnostic initiative. ICMR has recommended that every district hospital should have a blood culture or a urine culture. So improving availability of existing diagnostics and more research on having a point of care diagnostics. We are working with a private company to give us a typhoid test. So this is the way, identify priorities, and going after it will help us improve antimicrobial prescriptions.
Before that, there was a question by Harini. Any resources for detailed overview of diagnostic algorithms from bioinformatics and data science perspective?
Well, we will be developing the validation protocols. But from bioinformatics perspective, I don't think we will be developing anything. There are certain approaches which are being followed by some IITs and some other research institutions. Those investigators can probably help with that.
Then Dr. S.P. Swain, we don't see any funding for options for diagnostics. I'm sorry, for drugs, only for diagnostics. Well, that's a...
I have already answered that. This is a very, very, resource intensive area for development. But yes, we do fund small initiatives in ICMR also on new drug development. We make sure that we fund projects. They are uniformly divided into new drug development, antimicrobial stewardship, understanding the basic mechanisms. And I think BIRAC or CCAMP should be in a better position to fund new drug development through your CARB-X and other grants.
Yes, I think Dr. Swain is, if I'm not wrong, you're from NIPER, Dr. Swain, working. Okay, so Suraj also says, hello, ma'am, thanks for the wonderful talk. I am Suraj and have one doubt, ma'am. How do you see the scope of nanoparticles against AMR in future? Because both nanoparticles and antibiotics in the long term could be toxic when its use cross the limit.
So nanoparticles actually help us reduce the quantity of antimicrobial being delivered. So it was one of the areas which ICMR also funded. But unfortunately, what you have mentioned is the toxicity is something which is preventing us from using both these technologies together. I haven't come across a study which, because initially when nanoparticles was introduced, there was much hype around it. Maybe it can reduce, but then I don't think it is helping at the bug level to overcome the mechanisms of resistance. So that's, and plus added toxicity to it, it hasn't helped us go anywhere.
Debashish Banerjee, what according to you is the most promising area of therapeutics to treat AMR? AMPs, bacteriophages, naturopathy, or a combinatorial approach?
Well, combinations work very well. Carbapenem resistant patients are, organisms are sometimes treated very well with carbapenem, enhanced dose of carbapenem and colistin. This is what my clinician colleagues tell me. Bacteriophages, there is a lot of hype around it, but it continues to remain most of an academic pursuit, and I haven't really come across studies where they have been used on a large scale. There were few patients who received them, but then they were not more than, a few patients who actually benefited from this. Naturopathy is more of a prevention. I wouldn't say it can treat AMR. That's a statement that I would refrain from making, that naturopathy can treat AMR. But yes, that's why AMR is serious. If we don't have drugs, so many times I get phone calls from relatives of patients who have acquired highly drug-resistant Klebsiella pneumoniae or other such infections. And most of the time when you call back or inquire 2 weeks later that how's the patient doing, and they said the patient is no more. So that's why it is very important to, because we don't understand the extent of the problem. But the problem is huge. If the patient acquires Klebsiella pneumoniae, which is multi-drug resistant UTI, very difficult to treat that patient. So then doctors go back to repurposed drug, nitrofurantoin, or something like colistin is used, which was not used earlier. But then these drugs also have their own toxicity issues. So that's why antimicrobial stewardship is the answer. And for that, responsible antimicrobial prescription is the only outcome, unless we see more new antimicrobials coming in.
Zahid, as rightly pointed out in the talk, antibiotic treatment is cheaper than diagnosing antibiotic susceptibility. Plus, AMR diagnostics for animals seems a far cry. In such a scenario, how can government and the regulator change policy and incentivize AMR diagnostic innovators, particularly in the private sector? I think that also you have covered, but
yeah, AMR diagnostics are more expensive than the antibiotic. You can get an antibiotic for 100 rupees, but for a blood culture, you have to spend 500 rupees. That is why I highlighted that we need point of care diagnostics and rapid diagnostics. Just see how the cheap glucose strips or the RDTs for malaria have completely revolutionized the detection of these two diseases. So there is plenty to learn from that. There is just a point of care test available for typhoid, which is cheaper than, because the sensitivity and specificity is not so good. So that's why the treatment of typhoid is complicated. So we don't just need affordable tests, we need quality affordable tests for timely detection and treatment.
Then, of course, there's somebody called Galaxy Note 10 who is asking, what is the desired sensitivity and specificity for RDTs if we target multiplexing mode of detection?
Well, that will depend what you are detecting and what detection limit of detection is there. So there, I think that's why we need good target product profiles. Because once you have a TPP for a particular pathogen or a particular syndrome, for example, we are developing one for sepsis, which will have multiplex. But then how the sensitivity and specificity has to be adjusted, that has to be decided by the clinician, by the microbiologist, and by the statistician or epidemiologist who knows the local data. That's why I emphasize that TPPs are very, very important.
Anukrita wants to know, do you think drug development should also target boosting the immune system and not only treat the pathogens? Because in the long run, mutation rate will anyway be higher than novel
There has
antibiotic.
been research in this area. And I did hear a talk a few months back by Victor, I don't remember his last name. That was a beautiful talk on how they have augmented host genetic factors or host immunologic factors to actually fight the drug resistant Staph aureus. And this part is not being well researched, but then there is plenty of scope for understanding the role of host genetics and host immunity in augmenting the host response to fighting drug-resistant infection. This is some area which really needs to be developed. Host genetics is not very well researched for drug-resistant infections or for bacterial infections for that matter.
Then, okay. Ma'am, is there a way to control or contain the spread of AMR?
Don't have an antibiotic next time you have a cold or a diarrhea without knowing that it is a bacterial infection. That way you would be able to do your bit at least.
Your bit, yeah. Dr. Manish Paul, morning. How are you? Dr. Manish Paul, maybe you can ask the question yourself.
Sorry, I was answering Hari Vishal. Antimicrobial stewardship is the best.
Okay. Sorry, I thought.
Can I add a point to what Kamini was mentioning regarding the animal testing? If any, I am the co-inventor of the N-metazobactam, which has finished phase 3 trials now and in Europe, and we are going to launch the clinical trials in India now, the phase 3, before we bring it to the market. So anybody would like to discuss or know more about the animal testing, somebody asks a question, please do get in touch with me. I have journeyed from ideation.
I will also get in touch with you.
Absolutely. From ideation till the clinics, I have journeyed with this molecule and finished phase 3 successfully in Europe.
Maybe Nandita, you can have his talk next time.
We can do that, yes.
I will also attend it.
And Dr. Manish, if you can just share your email ID on the chat so that people who want to contact you can contact you.
Sure, absolutely.
Then does MDR TB diagnosis fall under purview of AMR work envisioned in this seminar?
Yeah, so AMR initiative of ICMR, which I look after, it doesn't have TB, because for TB there is a vertical program under Ministry of Health, and we only deal with the group of pathogens and infections for which there is no program. For example, E. coli, Klebsiella pneumoniae, typhoid, there is no program in ministry for that. So that's why the focus is to strengthen research in this area. TB essentially doesn't fall in my area in ICMR.
I think we've covered all the questions. If I've missed anybody, please unmute and ask your question. Most of them, Dr. Walia, are asking for your slides. Should be possible to share them?
There are 1 or 2 slides which are like unpublished data. So I would remove them and share my slide presentation with you, and you can.
Okay, then we can forward it. Yeah. Okay. So on that note, I think before we end, just wanted to let everybody know that we have, as part of the CARB-X GAN accelerator, just on the 15th launched a challenge. Small challenge for diagnostics, for any idea in diagnostics to, that can, for point of care and accurate diagnostics, to cover some of the priority pathogens, and particularly for that in the LMICs, in the low and middle income group thing. Maybe if I could request, I know this is a little bit, last minute, but if I could request either Swati or Meghna to just quickly share that poster, if it is.
I'll share, Nandita. Just give me a minute.
Okay. Yeah. So do apply for it, and I think we will get in touch with you.
And yeah, Nandita, we can have a quick group photo.
Group photo in the meanwhile, yeah, sure. Please do, all of you switch on your.
Dr. Kamini, I was also at Hopkins with Bill Bishai and Dr. Kim.
Okay, nice.
So I saw
How
your
long
background
you were
like
there?
2.5 years at Hopkins, 2003.
Okay. I was there from 2004 to 5, 1 year for MPH program.
Ah, you overlapped. Sanjay Jain was there in your.
I may not know. It was a long time back. And
requesting everybody to
please switch on the camera.
As many of you would like to, please do.
Nice.
Yeah, ready? All right. Yeah. Thank you.
Thanks, Chandrika. And Dr. Kamini, I really thanks a lot. I think it's always a pleasure to hear you and also read all your reports. I think it's, you explain everything so simply that even somebody who doesn't understand this space can very easily, grasp the whole issue. And I think that today's talk especially was really very interesting. Even though it was just 60 minutes or whatever the time, I think the way you covered the range of subjects and in such a simple this thing that it has been really, really well, I'm guessing well received because everybody wants your presentation. Thanks a lot. And yeah, we will look forward to working with you as well as with ICMR on seeing how some of these innovations can be brought, into the market, and so that they do reach their end use case is, becomes possible. So thanks a lot, and I'm sure everybody joins me in thanking you for this wonderful talk.
Thank you, Nandita. Pleasure to be
thanks.
associated with you.
Thanks.
Thanks.
Yeah. Bye.
Bye.